Welcome, forensic students. Mr. Mounty here. We are back with another little lesson on blood typing. Remember, blood chapter is broken up into two parts. Part one is blood typing. Part two is blood spatter. Today, we're going to continue with our blood typing analysis of our curriculum. So hopefully, you were able to take a look at Mr. Spears' video he posted having to do with the virtual lab, where hopefully you saved the patients. If you did not complete that yet, I recommend you kind of stop there and finish that virtual lab before you kind of move on as we go through our next topic in blood typing. So today we're going to recall back to our biology knowledge. And towards the end of biology year, when you took it, we started to talk about, or whichever class you had, started to talk about genetics. And in that genetics topic, you looked at Punnett squares. So we're looking at Punnett squares again to kind of realize how can a Punnett square help us in the field of forensics, and what kind of tell us as far as what potential blood types a victim or a witness or a suspect may end up having. So if you have your notes open, I recommend you do that so you can kind of follow along as I go through as well. So okay, so this is the kind of notes part. So in your assignment sheet, as you'll see, the top part is kind of notes and overview, which has to do with kind of background knowledge on biology. And then underneath that, you'll have the actual assignment. So the first part is just kind of review. So if you remember, A blood cells have A antigens, B blood cells have B antigens, AB blood has both antigens, and O blood has neither antigens. It's kind of just a review from the lesson that I posted last week. If you don't know what I'm talking about, then I strongly recommend you go back, review that lesson before we kind of move forward. Now, I'm going to be talking specifically about genes. So if you remember back to biology, your genes are made up of alleles, and you get two alleles. You get one allele from your mother and one allele from your father. And there's some properties that come with alleles. Remember, some properties, an allele might be a dominant gene or a dominant allele as opposed to a recessive allele. So to give you an idea, remember, dominant alleles are always expressed. That trait is always going to be expressed, as opposed to recessive alleles. Those traits are only expressed if it has two of the same recessive allele per gene. So to kind of give you an idea, if you look here, the potential alleles for blood type are IA, IB, and I. Now, the I stands for immunoglobulin, which is this term right here. Has to do with the white blood cell that would be triggered to attack, but I just kind of wanted you to focus on the A, the B, and the little I. So we'll use some abbreviations when we get to our guided practice, so I can kind of show you an easier way than having to do these little subscripts, especially on the computer. If we were writing, it'd be a lot easier, and I would ask you to do it, but in this case, we will do a little bit of a shortcut. So IA, you get that, if you have that allele, you'll end up having A antigens on the blood cell. Doesn't necessarily always have A blood, but you will have A antigens on the blood cell. And I'll get to that in a second. So IB, you'll have B antigens on your red blood cell. And little I means you have no antigens on the blood cell if you have that allele. Now, IA, dominant, IB, dominant, little I, recessive. So let's look at some examples of what your genotypes might end up being. Remember, genotype is the combination of alleles that you may have. Now, if you have type A blood, two potential genotypes you'd have. You'd either have IA, or IA, little I. Remember, little I is recessive. So even though it's present next to an A allele, a blood type A allele, the blood type A allele, which is dominant, will be expressed. So you will have A blood if you have two A alleles or one A, one O allele. Okay, and that will give you blood type A. Kind of the same thing for blood type B. If you look here, if you have, I'll zoom this in so we kind of see a little better. if you look at blood type B, it could either have two uh, B alleles or one B, one little I, or O allele. That will give you a blood type of B. Now, if you have one A and one B, remember both A and B are dominant. So in this case, you have codominance. You'll have codominance in this case. So you'll have both present, which gives us the blood type AB. Which makes sense. You have both alleles present on your red blood cell, or both antigens present on your red blood cell. And the only way you can have a type O blood is if you have both recessive alleles of little I, little I. Okay, let's just kind of overview of what the genotype sequence will be depending on what blood type you have. Well, how does that kind of help us make a Punnett square? Well, let's take a look. So we'll do some examples now. Hopefully you kind of remember. So one thing to add, as far as RH factor, that's not going to come into play for our Punnett squares. We're not going to worry about the RH factor in this case. If you're taking a college level biology class, that might come into play. But for us, our purposes, we are going to focus on just the two alleles of A, B, or the three alleles of A, B, and O. Okay, so for when we do our guided practice, instead of having to make these little subscripts, it gets a little bit of a pain when we're kind of do this on a computer. To make things easier, we're going to abbreviate. So to make things a little easier, the IA allele, I'm just going to put A. The IB allele, I'm just going to put B. And the little I, I'm just going to have O. Okay, make things a little easier as we kind of go through a Punnett square. So, well, what is a Punnett square? Punnett square is going to help us kind of get an idea of what the potential outcomes of a mother and a father, their offspring, the traits of their offspring might have. So, scenario one, we have a father with AB blood and a mother with type O blood. Well, if they're type AB blood, the only potential, genotype they can have, if we remember, is right here. One IA, one IB. So one A allele and one B allele. Now they're codominant, so both traits are present, which is why you have A and B antigens on your red blood cell. So I'm going to put AB for the father, and then O for the mother. Now, if you remember, for O, that's a recessive. So if I have type O blood, I must have two little O alleles. Again, we're just kind of abbreviating. Instead of using the I's, we're going to make things a little easier on us. Okay. So father's going to go here. Mother's going to go here as we set up our Punnett square. So when we're going through our Punnett square, if you remember back from biology, we combine rows and columns. So let's go across here. So this allele could have a potential O here. And then obviously the same with the other potential allele. And then the B allele could have an O, this O or this O. So what does that tell me? Well, my phenotype ratio, which is the trait, what are my chances of the traits that will pop up? Well, I have two traits here. And if we remember, what is the AO that could be present? Well, we remember back looking at our notes sheet. AO, which is right here, IA and little I, will give me blood type A. And that's showing up twice. So two potentials for A blood, and the same thing here. Two B's for the potential B blood. So therefore, it is a 1-to-1 ratio of the offspring being A blood or B blood. So the only potential offspring of these two people kind of joining together to make any offspring, their child, will either be a 50% chance of A blood, 50% chance of B blood. Does that make sense? So let's look at one more. Whoa, what is that word? We look at this. Father is blood type A homozygous, and the mother is blood type B heterozygous. What the heck does that mean? Well, if we remember back to biology again, homozygous and heterozygous tells us information. Remember, homo meaning same, hetero meaning different. Well, if I have blood type A homozygous, that means the two alleles are the same. So if I have blood type A, going back to my note sheet and looking, okay, which genetic sequence, which genotype is the same allele? Well, it's not this one. It has to be IA. So it's two A's. So father is type A homozygous, two A's. Now the mother is type B heterozygous. Well, we remember, again, back from biology because we love it so much. Heterozygous means the different. So if they're blood type B heterozygous, it's not this one. It has to be this one. So it's one B and one O. So we'll put that for the mother. So don't get too scared if you see a term you're not sure of. Just remember homo means same, hetero means different. It gives you an idea what the genetic sequence is going to be for the two alleles. So we'll set up our Punnett square, put those in. Father, mother, and then let's join them up. So this A could have this B, those two alleles. This A could have this O. This A could have this B. And this A could have this O. Well, what does that tell us? Well, the phenotype ratio, remember phenotype meaning the trait present. So what kind of blood could they have? There's two ABs and two A's. Remember AO, O being recessive, A being dominant, so it's blood type A. Gives me another 1-to-1 ratio. So 50% chance being type AB blood, 50% chance being A blood. So one thing to look at this in terms of the field of forensics, if you find a pool of blood at a crime scene and you test that blood and you find that blood to be type B blood, and let's say there's a missing person, and the missing person who is a potential suspect who's missing, obviously. And you want to find out, is this blood belong to that victim? You know, maybe we're looking for a dead body, not so much a missing person who's alive. Well, then you could get blood samples from that missing person's mother and father and determine whether it's possible that it's that person. So instead of actually trying to get a DNA sequence, which takes a long time, especially depending on where this crime is located, you can rule out the person potentially right away. So in this case, if I test my missing person's mother and father, father being AA, mother being BO, and I know that the blood I found at the crime scene was type B blood, I know it could not be that person. This is a perfect example of easily looking at a situation where we could be able to rule out a that's not the person we're looking for. We're looking for somebody else. So this would be class evidence. But remember, class evidence is still important because it could potentially exonerate someone or omit someone that's not someone we want to investigate. So there's kind of a little overview of Punnett squares and kind of reminder of the stuff you learned back in biology. You'll have access to this note sheet, so you can refer to it if you ever need to, or look at this video again if you need to. But other than that, I'll see you next time.